to this afternoon's lecture. It's my great pleasure and honor to introduce to you one of our youngest researchers at UCT, youngest and one of the most brilliant. And please, I know you don't like long introductions, but this time I'm going to insist. Dr. Ursula Rolding holds a doctorate in neuroscience from the University of Cape Town, where she currently works for the UCT Neuroscience Institute and the Division of Neurosurgery. Based at the Red Cross War Memorial Children's Hospital, her work combines the clinical and laboratory domains and provides insights that benefit patients with brain injury. In 2008, she received the Women in Neuroscience Award from the American National Neurotrauma Society. She has spoken at academic conferences in over 11 countries, and she has been the recipient of numerous conference awards. Dr. Rolding is a member of the International Tuberculosis Meningitis Consortium, and she collaborates closely with colleagues at the University of Cambridge at the UK, the Erasmus and Radboud Universities in Holland, as well as Johns Hopkins Medical University in the States. She has published close to 30 academic articles, and she has also included prestigious journals such as Nature and The Lancet, and she has co-authored a number of textbook chapters. Besides that, she also supervises numerous postgraduate students. She lectures in neuroscience and research methodology, and she teaches at UCT Summer School. In 2020, she's going to begin a prestigious fellowship with the Francis Crick Institute in London, which is awarded to promising young researchers. And in 2012, she will chair and host the first international conference on intracranial pressures and brain monitoring on African soil. Let us give her a big welcome.
Are you happy for the lights to stay on? Or does that make it a little difficult for you to see the screen?
A little bit.
It's a bit hot here.
It's either like this or it's pitch black. Are you sure? It's like this or pitch black.
Well, do you want to try it completely dark and then decide? Can we just see what the difference is like?
We'll be able to take notes. Two different shots. Just asking.
Let me just say something. The lecture is being recorded, so that you can always watch it again if you want, if you can't take notes, but you prefer the light on. It's up to you.
It's very hot if you've got no air conditioning and all the lights on.
Okay, so shall we leave the lights off?
Yes.
Yes, okay, great. Thank you so much. Thanks. Well, good afternoon, ladies and gentlemen. It's a great honor for me to be teaching at Summer School again this year, and I want to thank the organizers of Summer School for the invitation. And they've asked me to expand on one of the lectures that I gave last year, which essentially focused on how we begin to investigate and understand the brain when it's injured. But it's really important before we can even begin to talk about the injured brain that we lay a foundation about normal brain structure and function. So I just want to get a sense, how many people in the audience have been to one of my talks before? Okay, so a fair number, thank you very much. And so for those of you who have been to one of my talks before, some of the first part of the lecture might be familiar to you, but for the interests of those members in the audience who haven't been to one of my talks, I do just need to go over basic brain anatomy, structure and function, so that we can then all have a foundation for which we can then continue with the lecture. So this is a 2-hour lecture slot. I'll ask you about an hour into the lecture if you would like to have a break, and I will allow a 15-minute period at the end, so I will finish at about 2:45, so that there will be time for questions. Good. So I have this enormous privilege of being able to work with the organ which I think is really the most fascinating organ, and I get to work with the brain on a daily basis. And I I think it's the most fascinating organ because it has such a major role to play in the very rich and complex experience of human existence. And it is really the organ that facilitates so much of our activity, including the ability to learn, the ability to communicate, not only to generate speech in different languages, but also to understand that speech, to understand eloquence of speech, beauty of speech. It is what facilitates us being able to hear and take in what's in our environment, to see as well. It's responsible for generating movement, as well as for bringing in the sensations that the body receives, like touch, for example. It's enormously integral to our thinking and our feelings, so both our cognitive experience as well as our emotional experience. It's what we use to be able to acknowledge certain sensations, like pain, for example, whether that be physical pain or emotional pain. And it's enormously important to our sense of self. And this is very closely linked to memory. Memory is really what helps us to have an identity of self. It's very integral to that. Of course, that's not the entire story, but memory is really, really a big part of it. And apart from the fact that the brain is enormously important for us as individuals and our experience as individuals, it's also hugely important in the way in which we interact with other people and the relationships that we can establish in society, in the workplace, but also in terms of the work that we generate, innovation, drive, ambition, all of those aspects, our talents, these are really manifest through actions of the brain as well. And then, of course, it's not all about work and not all about self-consciousness, it's also our experience of the joy of life. And so for me, the brain is really the custodian of both our present and our past, and of course also our future. And so for me, it really is the organ that encompasses everything that we know of as life and existence as human beings. So over the life course, the brain undergoes various changes. Development starts obviously in utero when the fetus is still busy growing. And then during childhood up until early adulthood, late adolescence, the brain undergoes a really dramatic period of development and maturation. And so it's really during childhood that people talk about the brain being very plastic. And what this essentially means is that the substrate of the brain, which is really our brain cells, they are able to make connections with other cells, and in making those connections, establish networks. And those networks are really the foundation of things like our memories, speech, learning, cognition, executive function, emotion. So the ability that the brain has in early life to really make many, many connections and to be quite malleable in the connections that it makes is what makes a young brain so capable of taking in so much information, of learning, of growing, of developing. And that's why we talk about young brains as being plastic. Of course, by the time one reaches early adulthood, the brain has largely established the way in which it's going to function, and it has an evolutionary advantage that the brain goes through a period of maturation and development and then reaches the period where it's fairly stable. And that's a survival advantage. The brain goes through the growth it needs to for us to be able to survive in the world, and once we have reached a certain skill set, that is then what the brain maintains. And if it were to remain incredibly plastic and open to all kinds of change, it can make experience a little bit challenging. And then, of course, as one progresses through the lifespan, some decline in brain function can occur, but there's a tremendous accumulation and aggregation of wisdom and knowledge and insight and decision-making and perspective and things like that, that we see the older members of society are much stronger at than younger members of society. So throughout the lifespan, the brain has its many different strengths and advantages. But it is also vulnerable throughout the lifespan to various different pathologies and forms of damage. And that happens from the moment that an embryo is growing in utero. We see lots of children who are born with congenital anomalies of the brain. Children's brains are plastic, but they're also vulnerable while they're developing. If they incur an injury, if they have an infection like meningitis. Of course, there are other pathologies like brain cancer, for example. I think most people are not aware of the fact that brain tumors are the most common solid form of cancer in children. So it really is something that doesn't only occur in older age. Brain cancer is something that occurs across the lifespan. Similarly, the risks, obviously, of things like stroke. There are different kinds of stroke. We'll talk a little bit about them later on. And people at different points in their lives are vulnerable to different kinds of stroke and the implications thereof. And then neurodegeneration is something that occurs in the older population, but there's a lot of research to show that even in younger adults who have been exposed to certain injurious circumstances, we can even start to see neurodegeneration in those individuals. So essentially there's a lot that the brain has to offer, but it is vulnerable for different reasons throughout the course of life. So before we go into more detail about those vulnerabilities, I just want to go over basic anatomy. So this is a diagram of the brain, and as you know, we have a left hemisphere and we have a right hemisphere, and the brain is divided into different lobes. So we have the frontal lobe over here. Frontal lobe is an incredibly important lobe. It's involved in many aspects of our cognitive function, executive function, things like judgment, decision-making, problem-solving, social appropriateness in interaction, communication, personality. A lot of that resides in the frontal lobe, and particularly in the prefrontal cortex. Another very important part of the frontal lobe is this strip over here, which is called the primary motor cortex. So the brain is made up of what are called sulci and gyri. So a sulcus is really the groove, and a gyrus is the fold. And the brain is really very folded in on itself. And so the primary motor cortex is a piece of the frontal lobe. It's a gyrus at the back of the frontal lobe, and this is responsible for all of our motor activity. And the left side of the brain controls the motor function of the right side of the body, and the right side of the brain controls the motor function of the left side of the body. And different parts along this cortex are responsible for different limbs. One part's responsible for hands, another for arms, another for lips. It can be mapped out, actually, which parts are responsible for which limbs. Then we've got the parietal lobe over here. The parietal lobe has the primary sensory cortex, which sits directly behind the primary motor cortex. And it is the parallel of the primary motor cortex, but in terms of sensation. So while the motor cortex is responsible for sending messages out to the limbs and parts of the body that are involved in movement, the primary sensory cortex will receive information from those limbs, all those different parts of the body that we can then interpret. So sensation like touch, heat, pain, proprioception, all of those sorts of things are primarily controlled by the primary sensory cortex. And then the parietal lobe over here is involved in the entire experience of sensory perception, of spatial, understanding your body exists in space, but also understanding the spatial layout of areas, for example, and that's why it's also very involved in navigation. Then we have the most posterior lobe over here, which sits at the back of the brain. That is the occipital lobe, and that is the primary visual cortex. So it is responsible for receiving input from the eyes, and it processes that input, and then it sends a lot of that input to other parts of the brain. So as much as we talk about circumscribed areas that are involved with particular functions, the entire brain is involved in coordinating all of that input in a way that we understand it as a holistic experience. So there are a lot of parts of the brain, like the primary visual cortex is right at the back here, but what you can see is there's lots of additional tissue available in those lobes, and those are really what we call the association cortexes. So the association between having the vision of a safety pin, for example, or a needle, and what that means in terms of the pain associated with putting your finger on that needle is really about an association between the lobes that are responsible for the primary receipt of information. Then we have the temporal lobe over here. Temporal lobe is also enormously important. This is really where language is located, but language not only in the sense of being able to produce speech, but also being able to understand speech. So damage in one part of the temporal lobe might prevent a patient from being able to speak. Damage in a different part of the lobe might manifest in a patient who's very fluid and able to speak, but makes absolutely no sense in terms of what they're saying because there's the lack of comprehension. So every activity that we talk about is very nuanced, and there are multiple parts of the brain that are responsible for an overall function. Another important function of the temporal lobe is memory, and especially the inner structures of the temporal lobe, which we'll talk about next, enormously important in memory and in emotion. So again, on the inner surfaces of the temporal lobe, we have the limbic system, and that is the system that is responsible for memory, and it's very tightly linked to emotion. At the back here, which would be at the base of the skull, we have the cerebellum, very important for initiating movement, and controlling movement and coordination. So patients who struggle with Parkinson's will often have difficulties with the cerebellum. It's also very important for fine motor activity, whereas the primary motor cortex is more gross motor activity. And then, of course, the most important, one of the most important parts of the brain in terms of survival is the brainstem. And the brainstem is really what is responsible for controlling our key functions. And I'll talk a little bit more about that when I show you a cross-section through the brainstem. Okay, so now what this picture has done is it's removed one half of the brain so you can see the inside of the brain if we were to do a cut in this direction. And so these are the inner structures that I was telling you about. So here, for example, we have the hippocampus, which is involved in memory. We know, for example, that patients who have, so we have a hippocampus on the left side and on the right side. If a hippocampus needs to be removed on one side of the brain, that patient will still be able to form memories going forward. But if the hippocampi are removed on both sides, the patient will be rendered amnesic in terms of they're not able to develop any new memories. They can still access what memories they have from the past, but they're not able to develop any new memories. The hypothalamus, I'm sure many of you have heard of the hypothalamus. It's really the major regulator of hormones in the body, and it's often thought of as kind of the hormonal orchestra of the body. And it's a surprisingly very, very small, very thin part of the brain, but it controls all of our hormones, the release of hormones, the feeling of hunger or satiation, and it's very closely linked here to the pituitary gland, which participates with the hypothalamus to regulate our hormonal function. Then we have the amygdala over here. Amygdala is very important for emotion. And what you can see is that the amygdala and the hippocampus, they are quite closely linked with each other, and together with other structures, they form what's called an emotional circuitry, which is called the limbic system. And that's why it's not surprising that memories are so tightly linked to emotions, or that emotions can be very tightly linked to memories. And then what gets added to that are visual aspects of memory, sound, smells, sensations. These are all then linked into the memory system, and that's how, if you smell a perfume, for example, it might take you back to the days when you were a child and your grandmother used to hold you in your lap because it's the same perfume she wore. And it's because all of these areas are very tightly linked with each other. And that's why a lot of people will talk about brain function, lateralization, and localization of function. But the truth is, while there are certain parts of the brain that primarily do certain jobs, the entire brain is involved in the overall experience of whatever it is, whether it's emotion, memory, sensation, etc. Is anybody here familiar with the water systems in the brain, or familiar with the term hydrocephalus? Yes? Okay, so we have a ventricular system, so a water system in the brain. It's filled with a special kind of fluid, which is called cerebrospinal fluid. It's produced in these lateral ventricles of the brain. And essentially what that fluid does is enormously important for a number of reasons. It creates buoyancy. So the brain is actually quite heavy, but it needs to be held up on relatively thin neck and bony structure. So the CSF is like a buoy, or it allows the brain to actually rest in this water, and so it makes it much lighter. It's also very important for shock absorption. So the brain is incredibly vulnerable. We'll talk about that in greater detail. And that's why the CSF is also very important. It absorbs some of that shock, and so it can protect the brain to some extent. It's also continuous. The water in the ventricular system is continuous with the space around the cells of the brain. And so if drugs are going to be delivered to the brain, for example, they will often diffuse out of the blood vessels into the CSF and then travel to the brain cells in that way. And then recently, so the brain was always thought of being a structure that doesn't have a lymphatic system. So if you think about the rest of the body, we talk about the lymph nodes. So those lymph nodes are really, it's a different vascular kind of system that exists in the body that really clears things away. So the lymphatic vessels will clear away toxins, byproducts, etc., take them to the lymph nodes, and they will be metabolized or processed in whichever way they need to be. And so the brain was always thought of as not having a lymphatic system. But research in the last 5 years has actually demonstrated that the brain does indeed have a lymphatic system, and it's very tightly connected to the flow of the cerebrospinal fluid through the cells. And so the cerebrospinal fluid is not able only to deliver, was important for delivering substances to the brain, but it's also very important in removing substances from the brain. I will talk a little bit more about that because the cerebrospinal fluid is a major window for us as researchers and also for the clinicians to actually understanding the health of the brain, because it really bathes the brain cells and it gives us an idea of what's being delivered to the brain, but also what's being released by the brain cells. And we'll talk about that in a little bit more depth. So the CSF flows through the lateral ventricles down to the third ventricle, into the fourth ventricle, and then it exits, flows down the spinal cord, and then back up the convexity of the brain, where it's reabsorbed. And it's a constant cycle. And I'm going to show you an endoscopic video now. So this is a scope that is in the ventricle of the brain. This is the lateral ventricle. This is looking through one of the foramen into the third ventricle. This is the choroid plexus. Special kind of vasculature exists only in the brain. It produces the cerebrospinal fluid. This is the third ventricle. You've got your hypothalamus at the base, your mammillary bodies that are also a part of the hypothalamic auxiliary structures. And so this is essentially what that ventricular system looks like in the brain. In the next video, what the surgeons are doing is, if you have an accumulation of cerebrospinal fluid in the brain, that fluid needs to go somewhere. And we'll talk about exactly why that's important a little bit later. But when there's an accumulation of fluid, that's when patients will develop hydrocephalus, and it has some very severe potential consequences. So there are numerous ways in which the surgeons can actually try to divert that fluid. One way is they can actually put what's called a shunt. So it's a pipe that goes into the ventricular system, so through the brain, into the ventricular system, and then it gets tunneled under the skin, down into the peritoneal cavity, so into the abdominal cavity, where it can then be absorbed. And so the excess fluid essentially is shunted away from the brain into the peritoneal cavity, and it can be shunted elsewhere as well, into the lungs, around the heart, depending on the case. But then they developed a new technique where they are able to actually make a hole in the floor of the third ventricle, which allows the cerebrospinal fluid that is stuck in the ventricular system to actually communicate with the rest of the cerebrospinal fluid spaces in the brain, and so it can be absorbed that way. And that is called an endoscopic third ventriculostomy. So I'm going to show you a video here. So what they're doing is they're going to, and this looks enormous, but it's actually very, very small. They're going to puncture a hole through the floor of the third ventricle, which is what they're doing over here. And then what you'll see in the next part is that the fluid that was trapped can now actually communicate, and so you get that pulsation that comes back again. So the brain is actually constantly pulsating with the flow of cerebrospinal fluid if it is flowing as it should. So once again, a cross section, cerebellum over here, and this here is the brainstem. So I'm sure you've heard of people talk about the old brain and the new brain. So the old brain is essentially the brainstem over here, and it's called the old brain because it was the first part, or one of the older parts of the brain to actually develop. We share this brainstem with many other animals, and certainly also with non-human primates. And what we think of as the cortex, that very folded part of the brain, that is called the new brain, because that developed later. And it was really the advancement of that part of the brain that set us apart from other primates and enabled us to develop skills and expertise and abilities to communicate, develop societies, and innovation, all of those things that then set human beings apart and really put us at the top of the food chain. But this was really the most integral part of the brain for survival, because this is where the control of the heart rate and control of your respiratory rate exist. So, have you heard of the term brain dead? Right. So, brain death can refer to the fact that the cortex has been so significantly damaged that the person is no longer able to function as a human being in the way that we understand a functioning human being. Walking, talking, cognition, and those sorts of things. But they're still breathing, and their heart is still beating. And the reason is that the brainstem is, in those patients, still intact. So that will keep the heart going, it will keep the lungs going, but there is nothing going on above that. And that's when they refer to the patient as being brain dead. So they're not physically dead, the heart is still going, but there is no cognitive function of the cortex at all. So that's why this part of the brain is enormously important. And I'm going to come back to the vulnerability of this part of the brain when we start to talk about the dynamics in the brain, why we need to make sure that they're maintained in a certain equilibrium at all times. And one of the reasons is to protect this part of the brain. Okay, so this is just a picture where you can see the blue. That is the cerebrospinal fluid. So you can see essentially the brain is encased in cerebrospinal fluid, as is the spine. And that's why a lumbar puncture is done to access that cerebrospinal fluid, and then that can be tested to see whether the brain is infected or various other things. Are you familiar with the term meninges? Yes. So the meninges, you will know, are the protective sheaths that cover the brain. And there are three of them. The Dura Mater, which is the most outer layer. So we've got the skin of the scalp, we've got the skull bone, we have the venous blood, and then we have the Dura Mater, underneath which we've got the Subarachnoid Space, we've got the Subarachnoid, which is another meninges, and then on the brain surface itself, we have the Pia Mater. So Dura Mater, Arachnoid, and Pia Mater. So these sheaths are enormously important for protecting the brain, and the dynamics of blood around those sheaths is also very important. And we're going to talk about meningitis quite a bit later on, and that really involves the inflammation of those sheaths. But the particular form of meningitis that I'm going to talk to you about today is actually meningitis as a result of tuberculosis. And that is not only, our research suggests, an inflammation of the meninges themselves, but it actually involves disease of the entire brain. So a meningitis can be enormously dangerous because of the impact that that inflammation has on the rest of the brain. And we'll talk about that in a little bit more depth. So this is a video where we're now looking through that hole that they punctured in the third ventricle. And I want to point out, do you see this almost spiderweb-like appearance? That is the subarachnoid space, and it's called arachnoid and subarachnoid space because it looks like spiderwebs. So that's where arachnoid comes from. So once in the cerebrospinal fluid spaces around the brain, one has that kind of arachnoid appearance, and that's why it's called the arachnoid. And I always think it looks particularly beautiful, so I wanted to show you that. All right, so I showed you a picture of the brain where it was very nicely demarcated. This is the frontal lobe, and this is the parietal lobe, and this is the occipital lobe. But the truth is that when the surgeons open the skull, this is essentially what they're looking at. And it's not clear where the primary cortex is, where the sensory cortex is, or where the speech areas are. So there are a lot of skull landmarks that they can use to determine where certain things are. But then there are also a number of other techniques that they have available to them to be able to identify those eloquent areas that you really don't want to damage in a surgical procedure. And so sometimes also when people sometimes ask me the question, why are certain tumors inoperable, for example? Depends on the location in many respects. Because if it's located in an area, like for example the brainstem, if the surgeon goes in there and fiddles around, they'll kill the patient anyway. Or if they're located in an eloquent area of function, if they go in there and they take it out, the patient will be left with a disability afterwards. So exactly where things are located, and there's tremendous heterogeneity between individuals, is a very important part of surgical planning and figuring out how best to actually go about dealing with the brain if it needs to have surgical intervention. The other thing I want to point out is you can see that the brain is highly vascular. So there are big vessels on the surface of the brain, but there are also lots of vessels in the brain. So this is a kind of angiographic picture of the brain where you can see all of the arteries in the brain lighting up, and here you can see all of the vessels. So the arteries obviously are the vessels that bring oxygenated blood to the brain, and the veins are the vessels that take the deoxygenated or the used blood away from the brain back to the lungs, and then where they can be reoxygenated and then back to the heart that can pump it to the brain again and the rest of the body. So the brain only weighs 2% of the body's weight, but it requires 20% of the body's blood supply. And the reason is that the brain is a highly metabolic organ. And if you think about the degree of activity of the brain, as you're sitting here, much of your body is inactive, but your brain is always active. When you are being physically active, your body is active, but your brain is still active. And a lot of sleep research has shown that even during sleep, and especially during REM sleep, the brain is as active as it is during the waking state. So the brain is constantly busy, constantly working, and therefore needs a constant supply of blood, and it needs the blood because it needs oxygen and it needs glucose. And those are the two key substrates for energy metabolism, and that's really what the brain needs to keep itself alive. Okay, so which of the cells that make up the brain? I think most people know about neurons. Do you know about any others? So there are many other cells that are also incredibly important in the brain and that support the neurons. So we'll talk about the neurons first because they are the primary functional component of the brain, and they essentially look a lot like this. And they have a head that contains the nucleus where all of the genetic material is located and where the energy centers of the neuron will be located. And from the head of the neuron, you'll have all these outpouches, which are called dendrites. Then you'll have a long tail to the neuron, which is called the axon, and it has these little foot processors at the bottom, at the axon terminal. So when two neurons communicate with each other, the axon terminal, those little feet at the end of one axon, will connect with dendrites at the top of the next axon. And that's how a network is established. And if you look at how many dendrites there are present, you can imagine that on each of those points, this one neuron can make connections with other neurons, who or which can make as many connections with other neurons, and so it goes on. And so I heard a statistic that if you were to lay out the connectivity of the brain, it would reach the moon and back again. Because the brain has that much potential to make connections between cells. And that's why a pediatric or a child's brain is plastic, because it has still the capacity to make all of these connections with surrounding neurons. Whereas when certain networks are established in adulthood, once there's damage to that network, the neurons are not usually, in some cases or to some extent, they're able to compensate, but usually they're not able to re-establish connections with other neurons. Whereas in children, for example, especially in children who've had a disease in early childhood, so if they've had a very severe form of meningitis, for example, in childhood, and that has actually caused damage to half of the brain, as we saw in one of our patients, the entire left hemisphere of that patient's brain was largely not functional. And so that part of the hemisphere was generating a lot of seizures, that was causing a lot of damage to the brain, so they took that hemisphere out. So they essentially removed half the brain. And the patient was as functional before surgery as they were after surgery. And if you remember, I told you the left side of the brain controls the right side of the body. The left side is where the speech centers are located. The left side controls the sensation of the right side of the body, etc. But that patient, the brain of that patient had transferred its function from a non-functioning left hemisphere to the right hemisphere because it was still able to do that. So the brain is able to compensate a lot for as long as it still maintains plasticity, and that's the difficulty in adults. We lose that ability. Okay, so those are the key cells, but they are supported in their function by a number of other cells. We have microglia, those are the resident immune cells in the brain, so they scavenge away any pathogens, they clean away any debris. We have astrocytes, they form structural integrity of the neurons, they wrap their feet around the neurons. They're a very big part of the blood-brain barrier. Have you heard of the blood-brain barrier? Right, so you know that the blood-brain barrier is a very special vascular system that exists only in the brain, and it exists to prevent the indiscriminate influx of substances from the external environment into the brain because the brain is so fragile. So the astrocytes are a very big part of that as well, and they also can provide backup nutritional support for the neurons in cases where the neurons are not able to get sufficient blood and energy substrate, but also up until a point only. And then you have the oligodendrocytes. So the oligodendrocytes are enormously important for making the connection between neurons very fast. So the neurons you can think of like an electric wire, and we know that we put insulation around wires because it helps with the transfer of electric current. Now the communication between neurons is an electrochemical communication. Neurons will send an electrical signal down its axon and release a chemical which then generates an electrical signal in the next neuron. And that's how communication happens. And the oligodendrocytes wrap themselves around the neurons. They're very fatty cells, and they produce what's called myelin. So it's this white substance around neurons that helps them function better. And that's why we have grey matter on the external surface of the brain. That's where the neuronal cell bodies are. And we have white matter on the internal part of the brain because that's where the axons run, and those axons are myelinated and myelin is white. And then we have some deep-lying grey matter as well. Okay. Does anybody have any questions? That is basic anatomy, structure, and function. Yes?
Yeah. So that the axon, that you have got the dendrites, and then
Yes.
you've got the axon and the axon terminal.
Yes.
And if the axon terminal connects to another dendrite, is that the synapses?
That is the synapses, exactly. Yes. And that is when neurotransmitters are released. So when we talk about dopamine and serotonin, those are the chemicals that are released at that terminal over there, at the synapse. And that's why various medications will try to increase the amount of neurotransmitter or prevent the reuptake of it, etc. I didn't want to go into too much depth about that because I want to move on to the other things. Okay, can we move on? So, the brain is encased in this bony cavity, and it's really hidden from our view. There's no way we can see it, there's no way we can feel it, which is very different from other organs. For example, if the liver is swollen, you can palpate it, you can feel it, you can see it expanding. With the brain, it's completely a black box. We have no idea what is going on in the brain unless we find windows into the brain, and that's really what I want to talk to you about for the rest of the talk. So I mentioned that we have these vulnerabilities at different points in the brain, and a lot of the pathology of the brain boils down to similar principles. And so we're going to talk a little bit about what those principles are. And the way in which we can begin to get a window into the brain is first by looking at the brain just externally, various tests that can be done from the external environment. We need to start going more internally, more intracranially. So we'll talk about some of the ways in which we're actually able to tap into the inner environment of the brain. We need to look at the brain then at a cellular level, so from macroscopic to microscopic. And we get very microscopic, that we don't really look at it at a cellular level, we look at the components of the cells. So a component could be proteins, for example. And then now in the age of genetics, we are able to look at the very foundation of the structure of the brain, and that's really at a genetic level. Okay, so because the bone is in a fixed skull, it has space that is limited. So it has a certain amount of space for the cerebrospinal fluid, it has a certain amount of space for the tissue, it has a certain amount of space for blood. And that's both arterial and venous blood. So imagine that this is the brain and it has space for these components in a limited environment. So imagine if you have a brain tumor, for example. You suddenly have additional tissue that is present in a space that is already very limited. Or if you have hydrocephalus and you have an increased water component in the brain. Or if you have a stroke, for example, and you've suddenly had a vessel that's bled and there's all this extra blood that's in the brain, or you've had an injury, you've been in a car accident, you've had a skull fracture and it's bled underneath, again, there's all this extra blood in the brain. So what happens when these things occur? So the brain can compensate up until a point. So what it will do is, if you had, for example, increased cerebrospinal fluid, the brain can get a little squashed, and it can take in less blood and still maintain fairly healthy functioning. But it reaches a tipping point where it then can no longer function if it's not able to return to this equilibrium. So if you think about the pressure in the brain, it's not a linear relationship that as the pressure goes up, it goes up to the same degree, or the impact of it is the same. So it's a curve that looks like this, and I just want to explain this curve to you over here. So this is the volume in the brain, so kind of this component over here, and this is the amount of pressure in the head. So if you have a look over here at this point, at this volume in the brain, if you have that additional volume added, let's say you have a stroke, you've got this additional blood that gets added. This will be the increase in the pressure as a result of that additional blood. The brain can still cope. If the brain is reaching its tipping point, even a tiny additional bit of blood will push the pressure up significantly more, and the brain can't cope with that. So it can compensate up until a certain point. And one of our biggest jobs is to determine where on this curve the brain is. How much pressure can the brain handle in this particular patient on this particular day? And therefore, what do the clinicians need to do to control that pressure, minimize that pressure? And at how hard do we need to work to do that? So this is an example where you have a brain tumor, for example, you have this increase in tissue, and what happens is the brain starts to do what we call herniate. So the tissue starts to just get pushed out of the way. And what it will then do is it will eventually, when it's under so much pressure, it will actually just herniate through the entrance to the spinal cavity, and that is where the brainstem is located. So as the brain pushes down, the brainstem will actually push through that cavity, and that's instant death. Because you'll recall that I mentioned that the heart rate and respiratory rate are located in the brainstem. So at all costs, we have to avoid that from happening. So do you remember Liam Neeson's wife? About 10 years ago, she had a skiing accident and she was fine. She went home and she died. So she had an extradural hematoma, so that's blood outside the dural cavity. And what happened was that blood is under arterial pressure. So because the brain's able to compensate, she was okay. When she went to hospital, she seemed fine. But she went home and that bleed had continued. And she reached this point where the brain couldn't take it anymore, and her brain then herniated, and that's why she died. So establishing where the bleed is, how big it is, how fast it's going to grow, all of those sorts of things are enormously important in order to prevent this kind of phenomenon from occurring. But the same will occur if you have too much water in the brain. It will also herniate. So if it's too much blood, too much water, too much tissue from a brain tumor, any of those things can be detrimental. But that's the worst case. We have cases before that where an increase in pressure will mean that the first thing the brain will do is it will just stop allowing blood to flow into the brain. And as I mentioned earlier, the brain is hugely dependent on oxygen and glucose. Now it can survive without glucose for a fairly decent amount of time because it has got some reserve, but oxygen it needs and it's imperative. So the moment your pressure is high and your blood flow drops, your oxygenation drops, and that's when you get tissue death, and that's what we call an infarct. So similar with cardiac infarcts, where you have a heart attack, the cardiac muscle tissue dies, the same thing happens, the brain tissue dies, and then you get an infarct, but the problem with an infarct in the brain is there's nothing you can do about it, especially if you're not a child, and once that tissue is dead, it's dead. So this is also something that we absolutely have to avoid. So it's about avoiding death, but it's also about avoiding disability. So there are different kinds of strokes, and I know that stroke is something which many people have had experience with, family members, friends, somebody who's had a stroke. So what can happen in those cases is either you can have an ischemic stroke, which is where there is insufficient blood supply to the brain. That can be because there is a clot or a blockage of one of the vessels that supplies the brain. So all of the vessels above that clot will no longer be receiving blood. So all of the brain tissue that would be supplied by those vessels now no longer receive oxygen and they die. So that's why often patients post-stroke will be left with disabilities because parts of the brain have died as a result of a lack of blood flow. Hypertension, so patients who have had high blood pressure for too long, chronic hypertension, the vessels in the brain are under a lot of pressure for a very long time, and then various things can happen. You can have big vessels that the wall of the vessel starts to lose its integrity because it's dealing with high pressure all the time, develop an aneurysm, aneurysms burst, and you have a big bleed, and the part of the brain supplied by that vessel then dies. Or you can have it happening in smaller vessels, the smaller ones that go into the tissue, and then you can have smaller parts of the brain that infarct. The same happens with trauma. If you have a car accident or an assault to the head, you've got a skull fracture, you've got the bleed underneath. Again, it depends where the bleed is. It can be within the tissue itself, it can be on the outside, and so that can either cause tissue death or it can cause a lot of problems with pressure. So when we're trying to understand these different components of the brain and when we're trying to respond to them, there are various aspects of the brain that we can look at. The first thing we can look at is we can try to look at the brain's structure, so macroscopically. We can try to determine what the pressure is in the head and therefore respond to that pressure. We can try to determine how much oxygen is actually reaching the brain and can we improve that. And that's not only a function of what's happening in the brain, it's also a function of what's happening in the rest of the body. Because the lungs need to work to oxygenate blood. If we have a patient who's got lung pathology and the lungs aren't working, then they're not oxygenating the blood, and then the brain might be absolutely normal and fine, but a lack of oxygenated blood to the brain will then cause damage. Blood flow, as I mentioned, ensuring sufficient flow of blood to the brain is very important, but again that depends on the rest of the body. We need to make sure the heart is pumping at the right rate or the right amount to make sure that we're getting the amount of blood flow that we actually need, and therefore pathology of the heart, so heart disease, can have consequences for the brain. We need to look at the integrity of blood vessels. Blood vessels can become unhealthy for a number of reasons. We'll talk about some of those, so that's another thing that we can look at. We can look at the functioning of the cells. Are they undergoing cell death? Are they undergoing damage? Are they under attack? We can look at whether they are functioning properly by looking at their activity, by looking at the electrochemical activity of neurons. We can also look at whether they're actually producing energy, because only healthy cells will actually use oxygen and glucose to make energy. You can provide the brain with lots and lots of blood, but if the cells are dead or the cells are damaged and they can't use that, there's really no point. The tissue will die anyway. And then, of course, we can look at it on a genetic level. Okay, so I'm going to use tuberculous meningitis as a case. I think it demonstrates a lot of the principles of an injured brain that generalizes to other forms of brain injury. It happens to be my particular area of specialist interest. It's something that it's the most fatal form of tuberculosis. Most people are not aware of the fact that one can actually get TB in the brain, but it occurs very commonly still in TB endemic areas like South Africa. We see it quite a lot in children, and it leaves more than 60% of our patients with brain damage. We have quite a high mortality rate from TB meningitis as well. So it's a really very debilitating and very devastating disease, and so we're working very hard to try to improve the way in which we manage this disease, but we can only really understand how to manage the disease better if we really understand what's happening to the brain while it's under attack. So I want to give you a summary of what tuberculous meningitis is and what are the problems that it presents to us in terms of the neuroscience aspects of it. So as you will all know, TB is an infection of the lungs, but the TB bacillus is extremely, extremely old. They have traces of TB in Tutankhamun's mummy, for example. So it has existed for many, many, many years. And it has evolved faster than we can evolve in understanding it and treating it. So what happens is that in some cases, when it infects the lungs, it can actually break out of the lungs and it can gain access to the bloodstream. And then through the bloodstream, the TB bacillus can actually disseminate to other organs. And one of those organs is the brain. But you can get TB of the liver and TB of the spine and various other organs as well. But the TB meningitis is the most fatal form of disseminated TB, but it's also the most fatal form of TB overall. It causes more death than pulmonary TB does. So what happens is, once the bacillus enters the brain recognizes that there's a pathogen and that it's under attack, and so it immediately generates an immune response. And if you think about when you cut yourself, for example, and an immune response gets generated, and sometimes when there's bacteria, pus will form. So the same thing happens in the brain, and there's a generation of a very, very thick pus, and I'll show you a video of what that pus looks like in a moment. But essentially, if you have a look at the scan, do you see this very white stuff? That is, so do you see over here, this black? That is water in the ventricle, so that's cerebrospinal fluid. It appears black on the scan. Now, where all of this whiteness is should be black as well. Those are cerebrospinal fluid spaces. But instead of being filled with water, they are filled with pus. And what that pus does is it blocks the flow of the water through the brain because it's sitting in the water cavities of the brain. And so the water can't flow the way it's supposed to, so it just accumulates because the brain will just continue to produce it. And so then you get hydrocephalus, so the ventricles start to expand, and that starts to put a lot of pressure on the brain and pushes against the brain. And then the brain starts to look extremely squeezed. And what happens, of course, then is with the increased pressure, you get decreased blood flow. The second thing that pus does is it actually wraps itself around the major vessels that supply the brain. And so it causes inflammation in those vessels. And so those vessels either close or they can spasm. So when they spasm, they also don't allow blood flow to come through. So if you have a look, this is an angiography image in one of our TB meningitis patients. And if you have a look, can you see there's a big vessel here on the right side? But can you see it's missing on the left side? And that's because the pus and the pathology in that vessel is so severe that the vessels actually get that damaged, and so blood flow again is compromised. So you've got decreased blood flow from pressure, you've got decreased blood flow from abnormal vessels. So ischemia is a major problem in these patients, and that really is what drives these infarcts that we see in these children and in adults as well. So more than 60% of our kids will walk out of hospital with bits of their brains that are damaged. In some cases it will be just little bits, in some cases it will be very substantial. So how do we even begin to try to understand this? Well, one of the things we can do, if we look at just from an extracranially, so now we're not going into the brain, we're just looking from the outside. So if you think about ultrasound, Doppler, so Doppler they use obviously in ships and things like that, we can use that to actually detect the flow of blood in the vessels that supply the brain. So that's called Transcranial Doppler. So we can look at how much blood is flowing through those vessels, at what pressure is it flowing, is the pressure too high, are the vessels spasming, and that can give us an indication potentially for something that we can respond to. As I mentioned, oxygen, we really want to understand what's happening to the oxygenation in the brain. So one of the ways to do that are the two sensors that we can place on the forehead, and they send an infrared light through the skull, and that actually can pick up how much oxygen is present in the blood. But again, that is unfortunately quite limited because it can only assess the surface layers of the brain. It can't penetrate much more deeply than that. And so that's called near-infrared spectroscopy. This is a near-infrared spectroscopy data reading that I did in one of my patients. This was the brain oxygenation reading. The patient then had lumbar puncture. Lumbar puncture meant that we could remove some of the cerebrospinal fluid, so we could decrease some of the pressure, and then we immediately saw an increase then in the brain oxygenation afterwards. So this is a non-invasive monitor that we can use to give us some sense of how the brain is responding to various interventions. EEG, I think probably many people here are familiar with EEG. EEG really just looks at the electrical activity of the brain. When a brain is abnormal, it has very low levels of activity. So they will look at the wave forms. Or if a brain is fitting, then instead of the electrical activity being sequential, so one neuron fires, then the next one, then the next one, and so it goes on, you have a group of neurons that all fire together. And that's when the patient will have a fit and will seize. And there can be a number of reasons why people will have seizures, epilepsy, and various other things. But in tuberculous meningitis, we see our patients fitting because the brain is inflamed, the brain has got insufficient blood supply and oxygen, and the brain tissue is dying. So abnormal tissue can't produce normal electrical signal, and so we will often see seizures. So they can be very diagnostically useful for us in TBM. Then, of course, we can do imaging. This is something which many people in the room will obviously be familiar with. I'm showing you the oldest form of imaging, what existed before CT scan and other more sophisticated techniques. Essentially, in years gone by, what they used to do was they used to use an X-ray, and they would do a lumbar puncture, and they would inject about 5 to 10 mils of air into the spinal space. And the air would travel up the spine into the brain, and they would observe, that is a bubble of air over there, they would observe where does the air go. And from that they could figure out, okay, there's a tumor that's preventing the flow of the air from spinal space into the lateral ventricles. It's getting stuck maybe further down, or it's shifted over more to the right than it should be. So maybe there's a tumor on the left-hand side that's pushing the brain over. So that was all they had available. And you can imagine that was really quite crude, until there came a man, a South African, by the name of Alan Cormack, who actually did some work in the current Neuroscience Institute building, the building for the institute that I work for. In one of those rooms in that building over at Cortex, he developed the physics behind the CT scanner. And he later, he published that, and it was later taken on by another researcher, and he used it to build the CT scanner, and then together they won the Nobel Prize for medicine for developing a CT scanner. And the CT scanner revolutionized what one could see, not only in the brain, but other organs as well, because you suddenly went from a picture like this to pictures like this, where you could get a sense of hydrocephalus, you could get a sense of lesions that were present in the brain, you could get a sense of inflammation that was present in the brain, you could look at how things changed over time and evolved, how they responded to treatment, all that sort of thing. So the CT scanner really made an enormous difference in both diagnostics as well as treatment. And then CT angiography allowed surgeons to actually image the vessels of the brain as well. So these are some images which are courtesy of my colleague, Professor Alan Taylor, who's the endovascular specialist in our division of neurosurgery. So here's an example of a CT scan that shows a bleed in a patient. So then they can do some angiography. They can actually image the vessels on an individual level. They can actually reconstruct what the vasculature looks like in that patient and identify where the abnormalities are. And then, as was the case in this particular patient, they identified a small aneurysm, and then they were able to actually go in, they were able to determine where they needed to go, and they were then able to clip that aneurysm. So this is really the value that imaging has added both to diagnostics and treatment. Okay, then there's been a wonderful innovation in CT imaging, which is called PET, positron emission tomography. And essentially what PET allows us to do is we're able to inject a patient with a radio-labeled substance. And then they go for a scan, and the scan is able to detect the distribution of that radio-labeled substance in the brain. So this, for example, this is a patient at rest. And then what they did was they gave the patient radio-labeled glucose, and glucose, you will know now, important for energy production, and they gave the patient various tasks, and they observed where that radio-labeled glucose went. And they used that as a proxy measure of which parts of the brain are active for those different tasks, and therefore are involved in those tasks. But PET has opened the doors in the most terrific way in terms of developing new treatments, for example. So this is some data from a colleague of mine. She works on TB meningitis as well. She's based at Johns Hopkins, and she developed the first pediatric animal model of TB meningitis. So there are various animal models, but nobody had done a pediatric one. I work in kids. There's so much that we can't answer in patients that we can answer with animal models, so Liz and I collaborate with each other. And so this was some work that she published 2 years ago now, where they took an antibiotic that's incredibly important for the treatment of TB, and they radio-labeled that antibiotic. And then they did PET imaging on the animals to actually observe where does the antibiotic go in the brain, and are we getting enough of it? And what they found was that so you can see these bits that are lighting up in red. That's the radio-labeled antibiotic. But this bit that she has highlighted in yellow is a part of the brain where there is a lot of TB disease. And what you can see is that there is insufficient, if any, of the antibiotic that's actually reaching the diseased part of the brain where you need the antibiotic to go. So this is one of our challenges with treating patients is we give them drugs and you hope they're going to cross the blood-brain barrier. Once they've crossed the blood-brain barrier, you hope they've crossed at the right concentration. When they have crossed, you hope that it actually goes to the area that needs it. But up until now, we have no data to support that what we're doing in terms of drug treatment is actually having the desired effect. And so PET is opening the window for us to be able to actually trace where drugs are going. And are they going to the right places? Are they going in the right concentrations? How can we then improve the delivery of drugs? So what Liz's supervisor has developed is an incredible nanoparticle that we can actually conjugate a drug onto. And that nanoparticle crosses the blood-brain barrier with tremendous efficacy. And it's very targeted in where it goes in the brain. And so one of the things that they're working on is looking at how can we then take certain drugs, add them to that nanoparticle, and improve the delivery of the drug to the brain and also to the specific target site. So that's really, really quite a fascinating area of research. And the exciting thing is we just launched our own PET scanner at the Neuroscience Institute earlier, towards the end of last year, so about a month or two ago. So really, really exciting research is going to come out of that soon, not only in the brain, but for the rest of the body as well. So the CT scanner really, really adds a lot. But then there was a researcher by the name of Peter Lauterberg who took it further, and he used magnetic resonance technology to actually improve the pictures that we see of the brain. And he developed the theory behind it, and he submitted it to Nature, which, as you'll know, is the most prestigious scientific journal, and it was rejected. And he wrote that you could write the entire history of science in the last 50 years in terms of papers rejected by Science or Nature, because that work that he had rejected by Nature was the theory behind an MRI scanner. And an MRI scanner has taken imaging to yet another level, apart from the fact that it gives a much higher resolution than a CT scanner. So, for example, if I take you back, okay, so have a look at this image. It's quite grainy. You can't tell that much detail. And then you have a look at that. You can see that the degree of clarity of the image is vastly superior. It's not only in terms of the clarity of the image, but we know in TB meningitis, for example, that if we do a CT scan on a patient when they get admitted to Red Cross, and it doesn't look like they have any infarcts, it doesn't mean that they don't have any infarcts. The CT scanner just can't detect them while they are happening. It can only detect them when they're already there. And by that stage, there's nothing we can do about them. We would prefer to know that there's an ischemic evolving process that we can then respond to and prevent the infarct. And an MRI scanner enables us to actually see things while they're evolving. And there are so many different modalities that have been added to the MRI scanner that one can look at so many different things. It gives you such fine resolution in terms of understanding what's happening. So these are some images again from our TB meningitis patients where we can see infarcts much more clearly. We can see the angiography is much clearer also on MRI. We also MRI the spine. And the important thing is that, especially in children, MRI scanner is not associated with radiation exposure. And in some of our children who have hydrocephalus, for example, they have very complicated hydrocephalus. They can have up to 50 CT scans, in a 5-year period. And if you think about the radiation of 50 CT scans, it really works out to be quite a lot. And we have no proper sense of the long-term impact of all of that radiation exposure on children. But the MRI scanner presents no radiation exposure at all. And so it provides better images and safer images. Having said that, there are some things where CT scanner is still better, and certainly in resource-limited environments like ours and many other parts of the world where they see a lot of the pathology still, MRI scanners are very expensive. And so CT scanners are still the major form of imaging in most places in the world, and they still offer a tremendous value. to diagnosis and patient management. So I'm sure many of you have heard of functional MRI. So functional MRI is where you use a MRI scanner and you give your patient or your participant an activity, and you observe which parts of the brain light up. And essentially what it's doing is it is looking where glucose metabolism is highest in the brain. So what they will do, for example, is they will have, in this particular study, which I thought was just a bit of an amusing question, they wanted to see if there was evidence of a Christmas spirit network in the brain. So they showed some of their participants Christmas images and some of their participants non-Christmas images. And then they looked at which parts of the brain lit up in a group that saw Christmas images relative to those who saw non-Christmas images, and then they determined from the difference what is the Christmas spirit network in the brain. So it's a fun idea, but it's got a lot of limitations, which I'll talk about just now. But so it's used a lot in some of the psychology studies, for example, but it is also used by surgeons, for example, those surgeons who need to do operations which may involve areas where certain function is located. Remember I mentioned that a surgeon needs to know where the speech center is, for example, so that they don't destroy that center when they're on their way to a tumor in the vicinity of that. So they will often do, like, this is an example, you can see there's an obvious pathology there that needs to come out. So when they do the surgical planning, they will sometimes do a functional MRI for. They'll say, okay, we know that that tumor is located in an area that has the function of speech or reading or whatever, and they will give the patient tests before. They will see which parts of the brain light up when they do those activities, and then they will try to avoid those parts of the brain when they plan their surgical route. What they will also sometimes do is keep the patient awake, and they will open the skull and they will stimulate different parts of the brain with an electrode and observe whether patients are then still able to speak or not, and that will also give them an idea of whether that part of the brain is something that they can do away with or not. But while functional MRI has become a huge area of research and lots of people use it to study all sorts of questions, recently there's been data to suggest that there are a lot of false positives. So they did, in this particular study, they looked at Atlantic salmon that were dead, and they found signals, positive signals, using functional MRI on those fish. So clearly, there is a degree of error in the signal that occurs. And also, it's very reliant on the mathematics to take the signal, model that signal, and then come up with a conclusion. And so there's been, this particular study looked at about 40,000 functional MRI studies using 3 of the major mathematical models, and found that there were quite a large percentage of errors that were detected. So while it is certainly a valuable and interesting tool, it is limited, like many other things. And one of the reasons, of course, is it's a proxy. One's using the consumption of glucose as a proxy for activity related to a particular task. But it may not, in fact, be as a result of that task, may be due to other activities that are going on. And so there are various limitations that one always needs to be aware of. This is one of the newer techniques, which is also quite fascinating. What they can do is they can actually look at the diffusion of water molecules in the brain, and then they can model the track that those water molecules are taking, and in that way they are able to model the different connections between neurons throughout the brain. So you can see that as much as we talk about this part of the brain is responsible for this function and this for that function, those parts of the brain are all very heavily connected with other parts of the brain. So it's really a very holistic experience. And so one of the things that they're doing is they're looking at this kind of imaging in patients who have maybe been exposed to trauma of some kind, or children who've been exposed to alcohol in utero, and they look at whether this sort of imaging in your non-alcoholic alcohol-exposed babies looks different to your alcohol-exposed babies, and can that give us an idea of how the alcohol exposure makes organic changes in the way in which the brain is establishing networks. So that's called diffusion-weighted imaging and tractography. So I mentioned earlier that the pus that collects in the brain in the TB meningitis patients is such a major problem. So this endoscopic video I showed you earlier, I just want to show you again because I want you to compare this to the next video that I'm going to show you. So remember I mentioned that this is inside the ventricles where the cerebrospinal fluid is created, and we're going into one of the other ventricles over here. And this is the same location in a patient with TB meningitis. And so you can see that the ventricles look vastly different. They're not that beautiful pearly white. You can see that there's a lot of disease that's actually manifesting on the walls of the ventricles, these little studs that we suspect might be little tuberculosis studs. That beautiful flow of that pulsation of the CSF isn't present because the flow of the water is being impeded by the pus that is present. So this is a video done by Professor Figaji, who heads up the pediatric neurosurgery unit at Red Cross. And it was the first opportunity that many researchers in TDM had to actually see the pus that they've been researching and thinking about for many, many years. And it was actually to visualize it and to be able to see it with an endoscope really kind of changes the way we think about it, because we also realize that if the walls of the ventricles are so full of pathology, then perhaps this isn't just a meningitis involving the meninges, but it's something that involves more of the brain. How are you feeling in terms of taking a break? Shall we continue? It's 1:15. I want to finish in half an hour so that there's time for questions. Are you happy to press on? Okay. All right, so that was all the extracranial tests that I told you about. Now we'll talk about some of the intracranial things that we can look at. So one of the things that the surgeons will do is they will put a catheter. This is a little bolt. They make a hole in the skull, and they're able to put a screw in a little bolt, and then through that they're able to insert very, very thin catheters into the brain. And those catheters can tell us what the pressure is inside the head. I mentioned earlier that pressure is such a major characteristic that we really try to monitor. We try to monitor it continuously, so we have a sense of how the pressure changes from moment to moment in a patient, and therefore how do things need to be changed, should the blood pressure be changed, should the ventilation be changed, and so on. So it really gives high resolution information. And then similarly, there's also a brain oxygenation catheter. So there are two catheters, the pressure and the oxygenation catheter, that go through the bolt together. And then we use a software program that was developed by our colleagues at the University of Cambridge, which allows us to record the pressure and the oxygen every second. And then we can actually look at that data over time. So it gives us this wonderful window into what's happening in the brain in real time and how the brain responds to changes. So for example, this is where the blood pressure of this patient was increased, and we can see what happens to the pressure as a result and what happens to the brain oxygenation as a result. And in some patients, an increase in blood pressure might cause a decrease in intracranial pressure, but in many patients it will cause an increase in intracranial pressure, because each patient is different, and even within an individual patient, they can differ from one day to the next. And so this is really what enables the individualization of patient care and tailoring patient care to individual patients. Similarly with ventilator settings and various other components of how you're treating the whole body systemically that impacts on the brain. And I always take the opportunity at this point to share with the audience that we are the only pediatric unit that does this level of sophisticated monitoring in children, with other monitors that I'll tell you about as we move through the talk. We're the only unit that does this level of sophisticated monitoring in children in the world. And so you can imagine that without this information, we'd be pretty blind as to how to treat patients. So this is just other examples, again, of how we look at changes in various things, medication, ventilation, blood pressure, etc. Then I mentioned to you that we want to understand whether cells are able to actually use the oxygen and the glucose that's being delivered to them to produce the energy. So one of the ways you can do that is to actually look at the byproducts of metabolism of the cells by looking at what are the brain cells releasing, what metabolites are they releasing. And so there's a very thin catheter, it's the thickness of a capillary, also gets inserted into the brain, and it's called a microdialysis catheter, and at its tip, so at this bit over here, it has a semi-permeable membrane. So it's a membrane with small little pores, little holes in it. And so any substances, essentially, that are being delivered to the brain will also pass across that membrane into this catheter. And any substances that are being released by the brain cells themselves will also cross this membrane into this catheter. And then we sample the fluid in that catheter every hour. So that gives us an idea of what is the metabolic milieu of the brain cells, but also what is being delivered to the brain. For example, drugs, maybe you're interested, are the antibiotics being delivered to the brain, or is the brain releasing markers of injury, or markers of death, or markers of metabolism and so on. So we're able to actually sample that. This is what the system looks like, and we have a little vial that we collect that fluid in every hour, and so we use that as part of our research. All right, so we talked about some of the macroscopic things we can do, and we've talked about some of the, again, sort of larger intracranial things that we can do. But the truth is that we know that there are a lot of things that are happening at a cellular level. So this is from a publication that we did trying to break down some of the cellular activities that are going on in the brain of patients with TB meningitis. We know there are lots of cells coming into the brain, there are lots of cells in the brain that are doing things, they're releasing proteins, they're releasing enzymes, those proteins are doing things to the cells. There's a lot that's going on at a cellular level, and we really need to try to understand that. So we try to look at what's happening, what are the molecules that are present, that are being released, and what does that tell us about inflammation in the brain? What can that tell us about the injury of the cells? What can that tell us about the death of the cells? So for example, are the cells dying and releasing their components? And if we're detecting proteins that are present in the cell membrane, does that mean that the cell membrane is damaged and is breaking? If it's damaged and breaking, why is that happening? Is there a process we can respond to? Are there structural changes that are occurring in the brain? How are the cells functioning? For example, the metabolites that I mentioned will tell us how the cells are functioning. And we can also look at different molecules that are released by the pathogen. So we can look at, my research focuses on the host, so on the patient, but there are whole labs that focus on trying to understand the bacillus, the bug itself. And there's lots of chemicals and analytes that you can detect in human tissues that tell you something about the way in which the pathogen is actually operating. And these are what we call biomarkers. So biomarkers can be many things. They can be proteins, they can be enzymes, they can be cells, DNA, RNA. So many people will be familiar with biomarkers associated with heart attacks. For example, when a patient is suspected of having a heart attack, they will do a blood test. They'll look for a specific protein that comes from cardiac muscle tissue, and they'll be able to say this patient had a heart attack or they didn't. The heart attack was this severe. They'll maybe repeat that test in a couple of weeks' time and say things have improved. Or if a patient has cancer, for example, prostate cancer, maybe they will look again at markers in the blood that are markers of cancer. They will follow those. So biomarkers can help us to assess injury severity. They can help us to understand disease processes. They're very important for diagnosis. They can tell us quite a bit about pathogens as well, pathogen function, especially if they're biomarkers specific to a pathogen. If you're trying treatment and you're looking at those biomarkers and want to see how do those biomarker concentrations change on treatment, it can tell you about the response to treatment. And then if there's certain information that they can tell us about prognosis. But we don't have good biomarkers of the brain. And there's been quite a bit of research in traumatic brain injury, specifically looking at biomarkers of brain injury. But they've never been looked at in TB meningitis before, at least not in conjunction with looking at other markers of disease and inflammation and looking at a range of these biomarkers. So that's what we did as part of my PhD project. We said we know there's inflammation, and we know most researchers are focused on that pus and that inflammation, but does that inflammation actually correlate with the amount of tissue death and tissue injury that we see in these patients? So what we did was we identified 3 different biomarkers of brain tissue injury, and we analyzed them in our patients on admission in week 1, week 2, and week 3. We wanted to see how they changed over time. And we looked at them in association with markers of inflammation, because we know inflammation is a big part of the disease process. And what we found was that if we looked over time, the markers of inflammation, they decreased in all of the patients, which would suggest that the inflammation is responding to the treatment. But if you have a look here, you can see the patients in pink, right? This is time. So you can see the biomarkers of brain tissue injury, they decreased, you can see over here, in the patients in pink. And those are the patients who survived. But you can see there are patients here that I've identified in black. Those are the patients who died. And you can see that their markers of brain tissue injury continue to rise over time, despite treatment, and despite the fact that the inflammation was decreasing. So clearly, there are injury mechanisms that are ongoing in these patients, and we don't understand what those injury mechanisms are, and we don't understand how to respond to them. So when we think of those biomarkers, they are fairly downstream. They are endpoints of a process. And if we really want to understand what that process is, we need to have a look at the source, and that is really to have a look at it on a genetic level. So during my postdoc, that's what we did. We went and we looked at those same group of patients, but we looked at them on a genetic level. So I like to think of DNA and genes in this way. It helps me to bend my mind around them, so I'll share this with you. So DNA, you can think of as a language, and it is in fact. It's made up of letters. And the gene is the story that language, when you put it together, it makes a story, and that is the gene. So the genes are made up of pieces of DNA. And when you combine genes together, they make a volume of stories, which are our chromosomes. And all of those chromosomes get packaged into the nucleus in the cell, which is like the library of all of our genetic material. So what we want to do is understand, when we look at TB meningitis, we see holes in the brain. That's the end product. We've seen that there are markers of brain tissue injury that are increased, but what can we say about which genes are associated with that tissue injury? And if we can understand which of the genes are switched on or switched off, maybe we can actually respond and develop new ways of treating these patients. So this is a little animation just to show you how genes work. So here is our DNA structure. And the DNA really is king. The DNA decides everything. And so when it receives a message that the brain is under attack from TB, for example, the DNA will actually uncoil itself. It will open like this. And then a sequence that's almost identical, but somewhat different, is generated from that piece of DNA that was open. And that is your RNA. And your RNA is essentially, sorry, it's just replaying this. The RNA is the message that your DNA is sending to the protein factories in the cell, telling the cell what to produce. So those could be hormones, they could be enzymes, they could be proteins involved in immunity, they could be proteins involved in repair, in signaling, in various other activities. But this is where it is determined how the body is going to respond, and this is the message that then determines how the body responds. So when we study this message, we know what the DNA was telling the body at that time. And so that is called RNA transcriptomics. Essentially, we're just studying this message to understand how the body responds to TB meningitis. So this is what we did, and we found that the signature was all relating to brain injury, and one of the pathways that came through as being very upregulated, so the genes associated with this particular pathway were expressing themselves, was a pathway relating to neuroexcitotoxicity. So I mentioned to you earlier that the brain communicates in an electrical, chemical way, but sometimes it can become overly electric, and that causes a lot of problems because the cell is not able to recover, the cell starts to undergo damage, a damaged cell becomes increasingly damaged, it releases damage-associated molecules that then cause damage to other molecules, that damaged signal is then propagated onto the next cell, and so it can cause a lot of injury. And this is a pathway of injury that's been researched in traumatic brain injury, but it's never been looked at in TB meningitis before. And so this data suggested to us that this might well be a signal or a pathway that's activated in these patients that's potentially contributing to brain injury. And if we can identify what are the different components of that pathway, can we target something in the pathway to actually stop it? And can we, in that way, try to prevent the injury from happening? So this is just an image to show you, and I'm sure you can appreciate. Can you see that there's more yellow here and there's more blue here? So this is just telling us these genes in yellow are more present in our patients with TB meningitis than in our patients who don't have TB meningitis. So we always need to compare to make sure that this isn't just normal functioning, but we compare it to a group of patients who are non-diseased, so that we know that the signal that we are picking up is a signal associated with that disease, and then we can hopefully work to respond to that. So this is essentially the DNA that's broken down into its individual components. Sorry, it's the other way around, like that. So here's the chromosome, and these are all the bits of DNA that make up the chromosome. So I wanted to just mention this because there's a lot of questions around the degree to which the environment has a role to play. And so it's this question of nature versus nurture. Are we vulnerable to certain illnesses, or does the body respond in certain ways because we're genetically prone to respond like that? Or is it an environmental exposure that has somehow had an impact on our genetic material, and that's why we've responded? And the truth is that there is increasing evidence that it is a combination of both, but it's hard to say it's X% genes and X% environment. So what I just wanted to demonstrate here is there are other components on the DNA molecule, or that are involved in the way in which the DNA molecule is formulated into the chromosome, that will change or can change in response to various environmental influences. Now, environmental influences could be many things. I think people will think easily of, oh, could it be environmental exposure to carbon monoxide, for example? But it could also be exposure to drugs, or it could be exposure to foods or various other things. And so that is what's referred to by the term epigenetics. It's about trying to understand the degree to which certain external factors can actually change not the DNA itself, but rather the way in which the DNA might be expressed. And if we can maybe try to understand more of that, can that also help us to develop better treatments, to develop better knowledge about things to avoid or things to do, and so on. So that's epigenetics. And it is a burgeoning field, it's a new field. It's something we haven't looked at yet, but we certainly will in time. But this is a very complex area of research that's very much still developing, and there are lots of things we're still learning and finding out, and the technology behind this is also still very complex, very expensive, and that's part of what I'm going to be doing with the fellowship with the Francis Crick, is that they have a lot of the technology available for us to do these sorts of things. And so we're hoping that we're going to delve deeper into trying to understand what's happening. So this is, again, just a summary It's of a review that we wrote where we've said, okay, we know that the bacteria enter the brain. We know that there are a lot of chemical responses when that happens. We know that those chemical responses manifest at a macroscopic level in terms of pressure, blood flow, brain oxygenation, etc. And we know that those eventually manifest as brain tissue infarcts and brain tissue death, which leads to mortality or morbidity. So it's about putting together this entire picture so that we really understand the disease and we can potentially intervene at various different points in the disease process to finally land up improving outcome. Because it really is unlikely that focusing on just one thing on its own will make all the difference that we need. We need to understand it at its various different levels. The final test that I'll just mention, of course, is cognitive testing. So we do this in our patients where we look at the degree to which they're able to engage in certain activities which would be appropriate to their age, and that can help us to look at various components of function, locomotion, their interaction with people, their hearing, their speech, eye-hand coordination and performance. So this is one of our T meningitis patients. The guardian was happy for me to show the video. So he suffered quite a lot of he had a very severe disease course, and this was when we were doing his neurodevelopmental assessment afterwards to get a sense of how the tissue damage actually manifests in terms of day-to-day activities. And then, of course, for many different conditions in children and in adults, there are various other tests, including tests that look at IQ, memory, executive function, mental processing, reaction time, learning, and various other things. So that is a different level of also trying to understand and investigate the degree of brain damage and how that manifests in a person's level of function. So that's the end of my talk. As was mentioned earlier, I work for the Neuroscience Institute at UCT. This is a new institute. The building is actually being launched in March. That was just completed at the end of last year. And essentially, it's the first institute of its kind on the African continent. It's an institute that combines clinical service and research for patients who have neurosurgical, neurological, psychiatric, and neuropsychological difficulties. So we're a combination of clinicians and researchers who are working together to try to answer clinical questions using the research tools that we have available to really try to understand those questions better. It's a very exciting institute. We're just starting. There are lots of things that are planned for the institute, and a lot of excitement also from international funders, both philanthropic and academic. So it really is about saying, yes, Cape Town was the place of first heart transplant, but Cape Town can also be the place, that kind of rises to the very top of neuroscience research globally. So thank you very much. To work in this incredible institution at Red Cross Children's Hospital, where I'm based in the neurosurgery division, and this is the view from our operating theatres. So, apart from the fact that they're wonderful, amazing, groundbreaking, cutting-edge clinical procedures that happen there, there's fantastic research that happens there, and it's just a wonderful environment to work in. And I'm incredibly grateful that I get to be part of the neurosurgical team there. So we've got about 20 minutes for questions, actually. So the floor is open. Yes, sir.
I've been working in psychiatric hospitals in the 1960s and 70s, and of course that's archaeology maybe. But I just wanted to ask you something. In those days, they still very strongly believed in shock therapy. And there another branch, which I hope has also gone into the downward, was frontal lobotomy.
Yes.
What would you take on this now, with the knowledge that you have now?
So, when it comes to the lobotomies, I think that was a very dark time of psychosurgery, but there's been a very wonderful revival of neuropsychosurgery in a sense, and that's really with focused procedures that are done to very focused structures in the brain. So those can be where they maybe put an electrode into the brain, for example. One of our colleagues at Red Cross, Nicola Enslin, he's very involved in plasticity. So these are children who, due to various injuries, have developed cerebral palsy, and they've become very spastic. And or they have a motor abnormality where they actually can't keep their bodies in control. And so he's able to put very small electrodes into the brain that release a pulse at a very specific rate and frequency that can actually put a complete stop to those very strange movements that the patients experience. And it's life-changing, actually. Or they've also looked at it for various psychiatric conditions, obsessive-compulsive disorder, depression, and some others, where they may cause a little damage to a certain part of the brain that actually also is associated with tremendous relief in those patients. So we've definitely moved away from cutting out whole pieces of the brain or destroying the whole lobe, but there are various surgical things that can be done for both movement disorders and psychiatric disorders that are incredibly elegant and incredibly effective and really life-changing. So that history is hopefully behind us, and there's a new future ahead. Other questions? Yes, sir.
Hi, I'm Rodriguez. I used to work here at the biochemistry department for about 20 odd years. But I'm very interested in the brain function.
Yes.
I didn't study the brain function as such. But now I think I'm more interested in Alzheimer's studies.
Yes.
So the question really is, I just read an article some time ago of the danger or the risks of brain surgery, because as you said yourself, there's a blood-brain barrier, which is very, very important in order to prevent any foreign object contaminants, for example, entering the brain. And one of the things that has been found is that if you form nuclei, even in blood vessels, that's when you get clots being formed. And so in the brain, after a brain surgery, for example, there's always that danger, I thought, that when you close up the brain, that child or that patient will eventually develop Alzheimer's disease, because that is an accumulation of plaques and tangles. I don't know if you read about that. So what is the risks involved with open brain surgery? That's my question.
So you actually have a neurosurgeon sitting behind you, and I will defer to him to weigh in on this. But one of the things with so I think one of the things to always be aware of is when a certain condition develops, like a neurodegenerative condition, there are a lot of factors that could have contributed to that. So it's very hard, I think, in most cases, to be able to determine that there's one single thing that was the cause of a neurodegenerative process. Because we know that there are many things that can contribute to changes in amyloid beta and tau that lead to those neurofibrillary tangles and those plaques. That can be sort of your general health, cardiac health, genetic vulnerability, and various other factors. So I think to pinpoint one particular thing would require a very large sample size that is followed over a very long period where you've controlled for all those potential confounding factors. So I think that would be a big question to ask and a hard answer to come to, but I will refer to Professor Bigaji.
My question was really has there been any thorough study made to follow up patients who have had open brain surgery, and how long have they lived?
Well, I think...
Have they actually been healthy all their lives in terms of neurodegenerative diseases like Alzheimer's or whatever?
I don't know of any, and I think it would be incredibly difficult to do because you get brain surgery and you get brain surgery. You can have brain surgery for a tumor, you can have brain surgery for a vascular thing.
I'm talking about all brain surgeries where you see, as soon as you open the brain up, the brain is exposed to air, okay? And that is the problem because the blood-brain barrier is very, very important. That's why we've got a closed system. And so I'm saying is, are all brain surgery operations safe?
Well, I think that there's a lot of life-changing
Does it depend on the, for
outcomes.
example, does it depend on the hospital?
I think it depends a lot on the hospital and the surgeon, but I'll leave it to the surgeon to comment.
Yeah, so it depends on who the surgeon is. But I think it would be, so there's no clear correlation with Alzheimer's disease and any operation that some people
Yeah,
have.
so
So one of the problems with the blood-brain barrier is that you see with Parkinson's disease, it's a global blood-brain barrier problem. So operating as one sector of the brain doesn't necessarily use the whole blood-brain
the
barrier.
immune system can take
Yeah, so that's the one thing. But specifically with Alzheimer's disease, it's not particularly a blood-brain barrier problem, it's a deposition of protein. And even the deposition of protein, you don't know whether that's the primary problem or it's a manifestation of the problem. And it's likely a genetic trigger that's completely unrelated to any of the things that we understand that are causative. Now, of course, every week that goes by, you'll learn about. I think last week I read that if you slept 1 hour less, you increase your risk of Alzheimer's disease. but next week you'll read that if you slept 1 hour more, you increase your risk of Alzheimer's disease. And if you had 5 apples, you decrease your risk of Parkinson's. But if you had meat products, you increase your risk of Parkinson's. So people are largely disabled because they don't know what to eat. And that's the problem with I'm sure Dr. Robin mentioned this correlation versus causation. Because so typically what they do is they'll take 10,000, 20,000 people and they'll number crunch them with what are called variables or factors. I mean, you understand. And then the problem with that is the greater the number of the subjects, the higher the chances of you coming up with a completely random statistical association. And I think that's typically what happens. But no, there's no association coming back to the question. But that said, the best brain operation is probably one avoided. But if you've got something in your brain that's going to kill you within the next hour or within the next couple of weeks, versus a very vague, undefined, and possibly small risk 30, 40 years down the line, I think
especially if you're 70 years old.
you live a good life.
Yes. No.
Is there any benefit to having an MRI with schizophrenia?
I don't know enough about schizophrenia to be able to answer that question, but I think with any imaging or any investigation, I think that there should be a clear reason that the value of doing that procedure is there, because otherwise we could, one could go to the extremes in any condition in terms of in doing all kinds of investigations, whether that actually makes a material difference to the way in which a patient gets treated, I think is the major question. And I don't know enough about schizophrenia to be able to answer that question specifically. Yes?
Maybe that sounds a little bit dark, but with the organic biomarkers, can they also... Okay, let me rephrase my question. With, for example, depression or anxiety with the chemical imbalance within the brain, is that shown within organic biomarkers? Just curious or not?
Well, it can be, and it would depend. So, for example, with depression, there are various neurotransmitters that, like serotonin, for example, is associated with the sense of well-being and happiness and so on. And so a lot of antidepressants will target serotonin, especially sort of trying to keep it in the system for longer. So there would be potentially certain things that you could measure as proxies of those different activities that can give you information about sort of what is the chemical milieu and therefore sort of how to respond to it. But it can be difficult because the brain is really difficult to access. And so apart from those extracranial things we talked about, the imaging and the transcranial Doppler and so on, a lumbar puncture is an extremely invasive procedure. And people wouldn't easily agree to a lumbar puncture. It's very, it also has risks associated with it, so it wouldn't be indicated unless there was a very real reason to do it. And the difficulty, as the gentleman mentioned, because of the blood-brain barrier, it's not only the difficulty of substances getting into the brain, but it's also the difficulty of substances getting from the brain into the bloodstream for us to be able to take a blood sample and then say, this is what's happening in the brain. Because we've tried this with our brain injury biomarkers, that they, for example, have a particular half-life, so they only survive for a certain amount of time. So they need to diffuse from the brain into the blood, and then we need to sample it before that particular analyte has decayed. And that can be really, really difficult to do. So that's one of the real difficulties of trying to use organic biomarkers for brain-related conditions, because to get the samples to test for those biomarkers can be difficult, but they do exist in some cases.
Why is it so important to have glucose in the brain?
So glucose is one of the key substrates to produce energy. So the brain will break down that glucose to a certain structure, which is called ATP, and that is the unit of energy that the brain uses. And it needs glucose and oxygen to do that.
Can I eat glucose, or can I feed my brain with glucose?
So normal eating, that's why we eat to provide glucose to all of our organs, because all of our organs require glucose to produce energy. So not eating is a problem, because then the body lacks the substrates that it needs to produce the energy that it needs. It needs energy for activity, needs energy for healing, for repair, all those sorts of things. So just normal eating should provide the glucose you need. Yeah. Yes, ma'am, in the white or pinkish, or yes.
I have high toxicity of lead, arsenic and mercury.
Sorry, I can't hear.
Sorry, I have high toxicity of lead, arsenic and mercury, and that leads to anemia and low blood pressure.
Yeah.
Will that impact
On blood flow to the brain. So general low blood pressure, so the brain will be prioritized over all other organs. So those of us who do have low blood pressure, for whatever reason, we may feel tired and a little bit lethargic and so on, but the brain will receive the blood supply that it requires. If it doesn't, you'll faint, so that you lie flat and the blood will flow to the brain. So no, in general, it should be, your brain would probably be okay, unless there's some major reason.
So after a traumatic brain injury, why is drinking considered so bad?
So drinking alters your cognitive capacity, and I don't know if it at an organic level that it's necessarily bad, but I think if somebody has had a severe brain injury or they're left with some kind of cognitive impairment, you probably don't want to add to that cognitive impairment. That's
so
what...
like the general reasons, like it would be bad for your balance and that kind.
I would imagine so. I mean, I can't... Can you imagine an organic reason? No.
My brother had a brain hemorrhage when he was 53.
Yes.
And was living with a shunt. He's now 60. My mother is here with me, she's 93. How likely is it that he's going to get into his 90s with that shunt?
There's so many other factors that would contribute to his longevity. His genetics.